Chapter 5. The Connective Semiconducting Crystal. Man is equally incapable of seeing the nothingness from which he emerges and the infinity in which he is engulfed. Blaise Pascal, 1623 to 1662. Connective tissue has not been perceived by the research establishment as a glamorous research target. You don't read breathless reports in the Washington Post of research published in the American Connective Tissue Review. While you might read a story about research published in the American Heart Journal or Neurobiology, connective tissue has failed to generate the same level of excitement as other organs. This might be about to change dramatically. You can think of connective tissue as being a system of bags that contains and gives structure to each of your organs, coupled with a system of wires that hold all the bags together. The wires wind through the joints of the skeleton, which supports the structure of wires and bags, and your organs are encased in sheaths of connective tissue called fascia. The fascia surrounding muscles, called the myofascia, terminates in ligaments and tendons attached to the bone, connective tissue system, and collagen. The tendons are composed of twisted collections of collagen bundles, each composed of collagen fibers. Collagen fibers are composed of collagen fibrils, assemblies of molecules secreted outside of specialized connective tissue cells, called fibroblasts. Taken as a whole, the connective tissue system is the largest organ of the body. Yet the simplicity and ubiquity of the connective tissue system masks an important characteristic. Connective tissue fibers are arranged in highly regular arrays. There's a name for a highly regular parallel array of molecules. Whether it's in liquid or solid form, it's called a crystal. The collagenous molecules in which all of your organs are encased function as a system of liquid crystals. Crystals' highly ordered array of molecules are found in several different kinds of tissue, including the DNA and genes, the photosensitive rod and cone cells at the back of the eye, the myelin sheath of nerve cells, the collagen molecules that make up connective tissue, muscle tissue's densely packed molecules of actin and myosin, the phospholipids of cell membranes. This crystalline structure of the collagen molecules that make up your connective tissue has a remarkable property. It is a semiconductor. Semiconductors are not only able to conduct energy in the way the wiring system in your house conducts electricity very quickly from one point to another. They're also able to conduct information. Think of your high-speed internet connection. Besides many other properties, semiconductors are also able to store energy, amplify signals, filter information, and move information in one direction, but not in another. In other words, the connective tissue system can also process information like the semiconductor chips in your computer. Your connective tissue system is well suited for the task of conveying both energy and information because it connects every part of your body to every other part. Think of the communication possibilities of this structure. Every organ of your body is encased in the body's largest organ, which functions as a liquid crystal semiconductor in the form of the connective tissue system. Another property of connective tissue is that it is a piezoelectric substance, which when compressed generates electricity. The piezoelectric constant of a dry tendon, for example, is nearly the same as that for a quartz crystal. The the same kind of crystal the Ute Indians used to create light in their sacred rattles. Semiconduction in connective tissues. You can see why it's time for us to start getting excited about the connective tissue system. The importance of its conductive properties is hard to overstate, for it explains one of the enduring mysteries of acupuncture. How stimulating one acupuncture meridian point, for instance in the ear, can have an effect on another point, for instance in the spleen. Mei Wan Ho, a researcher who has published several papers on the crystalline nature of connective tissue, says liquid crystallinity gives organisms their characteristic flexibility, exquisite sensitivity, and responsiveness, thus optimizing the rapid, noiseless intercommunication that enables the organism to function as a coherent, coordinated whole. Intercommunication also explains how tapping one part of the body, which creates a piezoelectric signal, then creates an effect in a distant part of the body, or in the entire body. Electrical signals can be carried throughout the body by the connective tissue. James Oshman, Ph.D., whose fascinating book, Energy Medicine in Therapeutics and Human Performance, assembles a great diversity of research from many disciplines into an understandable whole, says signals generated by the piezoelectric effect are essentially biological communications that inform neighboring cells and tissues. The fully integrated body may be a body that is entirely free of restrictions to the flow of signals. Cell biologists studying collagen reductively would miss these unique properties. Broken down into individual collagen molecules, connective tissue does not have the same characteristics, and it takes assembly into a parallel array structure to produce its ability to conduct and store energy. The electrical properties of the connective tissue also explain how cells can communicate much faster than the speed of neural transmission. Speaking into cells. Each cell also has a skeletal structure. The shape of the cell is partially determined by a system of cylindrical protein beams called microtubules. Microtubules are the girders of our cells. They are rigid protein structures, the rebar that give cells their shape. The unglamorous role of being merely a member of the supporting cast has resulted in less attention being paid to the microtubulars by researchers who've had much more glamorous cell structures, such as the mitochondria, the energy generators, the genes repository of blueprints, and the cell membrane letting signals in and out, to occupy their attention. Being a piece of rebar in a building is unlikely to attract as much attention as being the facade, the power plant, the lobby, or the elevators. We think rebars, and we automatically assume rigidity. But microtubulars do indeed provide cells with the property of rigidity. One of the overlooked properties of microtubulars, however, is their transience. Microtubulars are transient in the sense that they are rebuilt often, in some cells several times an hour. Far from being structures that are created during the division of a cell and which remain firmly in place throughout the cell's life, microtubules are rebuilt often and quickly. In the June 2004 issue of Science and Consciousness Review, John McCrone writes this about the microtubules of the brain. Do you know the half-life of a microtubule, the protein filaments that form the internal scaffolding of a cell? Just 10 minutes. That's an average of 10 minutes between assembly and destruction. Now, the brain is supposed to be some sort of computer. It is an intricate network of some 1,000 1,000 trillion synaptic connections, each of these synapses having been lovingly crafted by experience to have a particular shape, a particular neurochemistry. It is, of course, the information represented at these junctions that makes us who we are. But how the heck do these synapses retain a stable identity when the chemistry of cells is almost on the boil, with large molecules falling apart nearly as soon as they're made? The issue of molecular turnover is starting to hit home in neuroscience, especially now that the latent research techniques, such as fluorescent tagging, are revealing a far more frantic pace of activity than ever suspected. For instance, the actin filaments in dendrites can need replacing within 40 seconds, and the entire postsynaptic density, PSD, the protein-packed zone that powers synaptic activity, is replaced molecule for molecule, almost by the hour. The conclusion of this research is that the entire brain is being recycled about every other month, opening up an enormous field of inquiry into how neurological change interacts with changes in energy systems. The very skeleton of each cell is being revised several times each hour, as though a building's architecture was metamorphosing each day, far from the role of the passive supporters to which microtubules have sometimes been assigned. It turns out that they are active daily shapers of the very physical dimensions of a cell structure. Cutting-edge scientists are left wondering not so much how we can change, but how we can endure. Yet, this is only the beginning of our reevaluation of a microtubule's role, for it turns out that their reformulation of the structure of the cell is not random. Microtubules appear to be attuned to the energy field in which the cell exists. Not just mere girders, microtubules may in fact also be antennas, receiving signals from the environment and carrying out their complex restructuring in response to these signals. The cytoskeletons of cells are also piezoelectric conductors. Electric fields change their shape and mechanical stimuli their electrical condition. The role of the hollow core at the center of a microtubule is unknown, though some researchers have speculated that superconductivity and electromagnetic focusing take place in the cores. Cell biologist Bruce Lipton points out that conventional medicine works with the iron fillings, whereas a deeper form of healing would attempt to influence the magnetic field. Most doctors don't see the field, so they're trying to figure out the relationship between the fillings without even trying to incorporate the energy field in which they exist. Microtubules may be a key resonator with the energy field, and some researchers have suggested that they may play a key role in receiving information required to structure cells from the orchestra conductor of consciousness, anchoring cells to the connective system. An important class of protein sugars, called integrins, convey information between the environment outside the cell and the inside of the cell. They anchor the cytoskeleton and other internal elements of the cell structure, even the nucleus, to the connective tissue outside of the cell. So there is a feedback loop of information going into the cell and information emanating from the cell and being conveyed into the connective tissue matrix. The implications of this connectivity are staggering. It turns out that the internal structures of cells are also influenced through the connective tissue. with the energy environment we are creating with our thoughts and feelings. It's not just DNA that's being affected by our internally produced environment. It's not just our collagenous tissues. It is the very microtubular structure of our individual cells. Our bodies are more plastic than we once thought, and this flexibility carries with it the possibility of rapid, miraculous healing. The chemical signaling mechanisms of the nervous system communicate at a speed of between 10 and 100 meters per second, depending on the diameter of the axon. This is much too slow to explain many human processes. The neural signaling mechanisms described in most medical textbooks today are inadequate to explain, for instance, how a batter can hit a baseball at a speed of 50 meters per second. There's simply not enough time for the visual image of the ball leaving the pitcher's arm to travel from the batter's eye through the optical nerve to the visual cortex, and then to the centers of the brain that govern volition, then to those that generate muscle movement in the upper body, then down to the muscles themselves, instructing them to swing. There's only about 2/5 of 1 second between the ball leaving the pitcher's hand and the completion of the batter's swing. Neural signaling cannot do the job of conveying such a rapid response. Baseball catcher Tim McCarver rightly exclaimed, "The mind's a great thing as long as you don't have to use it." This is just one of the many functions in higher animals that cannot be explained in terms of capabilities of the neural signaling system. Each cell is going through around 100,000 chemical processes per second. These processes are coordinated among a body, a community of cells, that is composed of trillions of cells. Such coordination borders on the miraculous. How do these cells know how and when to create exactly the same reaction at exactly the same instances? In the field, Lynn McTaggart asks, if all these genes are working together like some unimaginably big orchestra, who or what is the conductor? And if all of these processes are due to simple chemical collision between molecules, how can it work anywhere near rapidly enough to account for the coherent behaviors that live beings exhibit every minute of their lives? Neural signaling cannot even begin to grapple with the cybernetic task of this magnitude. Rapid cellular signaling. If, on the contrary, we assume electromagnetic signals passing through a liquid crystal semiconductor, encasing all the organs with the microtubules in individual cells, resonating in symphony, many such processes suddenly make sense. Oshman sees the connective tissue system as a living network that corresponds to the operating system of a computer, enabling all the parts to work together smoothly. Joey Jones, Ph.D., and his research associates have tested the results of stimulation of the vision-related meridian points in the foot using a fMRI machine. They discovered that when these points are stimulated, neural circuits in the brain's occipital lobes are activated almost immediately at a rate far faster than neural conduction can explain. In the real world, our bodies function with an integration of stimulus and response at a speed much faster than that of neural signaling. A group of researchers led by Andrew Ahn, MD, of Harvard Medical School, looked at the possibilities that segments of acupuncture meridians that are associated with loose connective tissue planes between muscles or between muscle and bone, had a greater electrical conductivity than non-meridian points, and demonstrated this to be the case in a series of experiments published in 2005. Signals travel through these electromagnetic conduction pathways in the meridians at a pace many orders of magnitude higher than the signals traveling through the neural net, using a process called ElectroConformational Coupling. They can then influence protein folding. Just like my brain sending a signal to my body, I can send a signal to another trusted and functional vehicle, my car, when I want to unlock it and drive home. I can do this in one of two ways. One of them takes a long time, and the other occurs instantaneously. Attached to my key ring is a car key. I can insert it in the car door lock and turn it. Then open the door, reach inside to the other doors, and pull up on the doorknob to unlock them too. Then I can insert the key in the ignition, turn it on, and start the car. Now the process takes me about 6 seconds, and is accomplished by a physical, mechanical signaling system. I can also use the other element attached to my key ring, the automatic door opener and engine starter. One click and presto, the doors are unlocked and the engine starts. It takes a nanosecond to start the process, which is accomplished via an electromagnetic signaling system. Another example of a physical signaling mechanism is a letter. I can seal it in an envelope and drop it in a mailbox, and a mechanical system of trucks, hands, and conveyor belts will deliver it to the addressee in a few days. Or I can send the same message using an electronically based system. I can write the same letter, then simply hit the send key on my email program. The electronic communication reaches the recipient almost instantaneously. Psychologist David Feinstein, Ph.D., says electromagnetic frequencies are 100 times more efficient than chemical signals such as hormones and neurotransmitters in relaying information within a biological system. A calculation based on research conducted in the 1970s by Oxford University biophysicist C.W.F. McClare. This is not terribly surprising when you consider that many of the body's regulatory chemicals travel less than a centimeter in a second, while an electromagnetic wave could have traveled 3/4 of the distance to the moon in that time. Your body has both systems, a mechanical chemical signaling system based on the movement of charged ions across cell membranes, and the diffusion of hormones and neurotransmitters, and an electromagnetic signaling system. Both the mechanical and the electromagnetic system can activate cells and genes to accomplish the intent of the user. Yet current medical research focuses almost exclusively on the former and gives an inexplicably small amount of attention to the latter, and conventional medical treatment protocols practically ignore the much faster and more efficient energy signaling system. A field of electromagnetic radiation as the cause of the structure of the body and its cells was first postulated by Russian scientist Alexander Gershwin as early as the 1920s. In the same decade that the energy field of the heart was being mapped by Wilhelm Einthoven, and the EEG was being developed by Hans Berger, also in the 1920s. Researcher Elmer Lund at the University of Texas found that he could reorganize the cellular structure of small animals by applying an electric current strong enough to override the creature's electromagnetic field. Later studies by Herbert Froelich of the University of Liverpool predicted that liquid crystalline phospholipids just below the cell membrane. Vibrating at certain frequencies, now called Froelich frequencies, could synchronize activities between proteins and the system as a whole. He showed that at certain energy thresholds, molecules begin to vibrate in unison until they reach a high level of coherence. The moment molecules reach this state of coherence, they take on certain qualities of quantum mechanics, including non-locality. They get to the point where they can operate in tandem. The semiconductive properties of the connective tissue acting in resonance have a speed and power that far transcends other signaling mechanisms. Mei Wan Ho says that the crystalline structure of tissues and organs results in harmonic resonance of the entire structure. When the coherence builds to a certain level, the organism behaves as a single crystal. A threshold is reached where all of the atoms oscillate together in phase and send out a giant light track that is 1,000,000 times stronger than that emitted by individual atoms. This quantum coherence means that signals are passed back and forth in a continuous feedback loop from cells to brain to tissues in virtually the same instant. Communication is a two-way street. Not only does the environment of our thoughts, feelings communicate itself throughout the body via our liquid crystal semiconductor system, but changes in the state of our semiconductor system communicate themselves instantly to our consciousness. Our brains are receiving information from all over our bodies rapidly and continuously, and using this information to shape our choices, both conscious and unconscious. Physiology and consciousness are in an ongoing and inseparable feedback loop. Rather than being two separate processes, Oshman calls this the living matrix, and summarizes by saying, the connective tissue and cytoskeletons together form a structural, functional, and energetic continuum, extending into every nook and cranny of the body, even into the cell nucleus and genetic material. All forms of energy are rapidly generated, conducted, interpreted, and converted from one to another in sophisticated ways within the living matrix. No part of the organism is separate from this matrix. The resonant nature of the living matrix also suggests that it is possible that our connective tissue system may be a quantum resonator, conducting signals from the quantum field of the universe into the body and from the body to the field. The signals from our brains are communicated constantly via the matrix of energy conduction within our bodies. Every thought you think is echoing through your connective tissue communication system, turning genes on and off, producing either stress responses or healing responses. This understanding opens up a vast, new panorama of potential self-healing.